Hello, everybody. I am Carina Eklund, and I will talk to you about the quality assurance of HPV screening, laboratory audits, and proficiency panels for HPV testing. Why do we want to do laboratory audits? Since HPV-based screening is recommended as a public health policy in many countries, it has been important that the quality assurance of routine HPV laboratories analyses are done. Well-structured audits can systematically identify errors and analyze artifacts. It assesses the quality of HPV analysis as used in the screening lab by repeated estimation of the clinical sensitivity to identify women at risk for CIN3 or worse. This is important in the post-vaccination era to monitor which types of HPV that continue to cause disease in the population. And how can we do this laboratory audit? The same audit strategy as is presently used for cytology can also be used for the HPV testing. LBC samples that precedes the CIN3 diagnosis should be abnormal by a screening test. This is the basis for estimation of diagnostic sensitivity. We use a laboratory database on histology diagnosis to find the CIN3 cases and the cervical samples taken before diagnosis that should have been identifying the CIN3 case. As I said, the endpoint of this audit is the histopathologically diagnosed CIN3 biopsy. And we limit the audits to women with cervical LBC samples taken within 2 years prior to the CIN3 lesion. The CIN3 lesion should be diagnosed as a result of LBC screening for cervical cancer. Women who have been identified as CIN3 patients by detecting symptoms are not included in the audit. If no original HPV screening data is available, the LBC samples is retrieved from the sample archive and HPV tested during the audit. The pre-diagnostic cytology slide is also reanalyzed if the original diagnosis was normal. We performed an audit in our laboratory in Stockholm during 2013-2014. And in this table, you can see the analysis of the HPV analysis on the LBC samples on Cobas 4800 compared to the cytology to identify women with subsequent CIN3 in the biopsy. And as you can see, the Cobas identified 97% of the women as true positive, and it was 6 women or 3% that could not be identified with the Cobas HPV test. And the cytology diagnosis was almost as good. It identified 94% of the women as CIN3 cases, but it missed 11 women or 6%. This shows that it's important to perform a laboratory audit to see that your HPV test is working. If you don't get a good result in the audit, you have to consider to take measures to do the better HPV testing. Now I will talk a little bit about HPV global proficiency panels. We started to do HPV global proficiency panels that should be able to detect multiple infections. They should have a high capacity to detect typing error, and it should be possible to use with all HPV DNA tests. Our HPV global proficiency panels detects 14 high-risk types and 2 low-risk types. It contains 42 samples with plasmids, 1 negative control, and 3 extraction controls. This is how the proficiency panel looked in 2014. We have been issuing proficiency panels during 6 years, starting in 2008. All have been rather similar, but with a different content of the mix of the samples. As you can see, all the samples are included in 2 different copy numbers. For HPV 16 and 18 has a lower copy number. And then also there are 4 mixes with 4 HPV types in each mix. And we also have 3 samples that contains only HPV positive cell lines to see that the extraction process is working correctly. And the requirements to be proficient in our study, it's higher for the HPV 16 and 18 with detection of 50 international units in 5 microliters. The other HPV types require 500 genome equivalents in 5 microliters. But all types must be detected in both single and multiple infections, and no more than one false positive result is allowed. That is the same as 97% specificity. In 2014, the HPV panel was distributed to 24 laboratories in 6 different WHO regions. We did receive 148 datasets back with results from 121 laboratories. And laboratories that participate in testing these proficiency panels are both from clinical and research laboratories, it's kit manufacturers, and vaccine producers. And this shows the distribution of the laboratories that participated and submitted the results. And as you can see, Europe is highly overrepresented, but there are also quite a lot of laboratories from Southeast Asia and some from South America, and also one in Africa in 2014. This table shows a lot of information. The important thing here is to focus on the non-proficient tests. Non-proficient means that you have more than one false positive result. And as you can see, there were 31 assays reported to have more than 2 false positive results. And what is also important is that some of the assays, like linear array and HPV direct flowship, have a few laboratories that shows not proficient results, but there are also a few laboratories, or the same number of laboratories more or less, that has more than 100%, or that has 100% proficient result. So the same assay can work perfectly well in one laboratory and give a not proficient result in another laboratory, which means that it's not just the assay that is important, but also how you perform the assay. You can also see that there is a big variety of different assays. The ones that are labeled gray here are the commercial assays, and there are also quite a few laboratories that use in-house assay. This picture shows the sensitivity by HPV type. And this is to illustrate that it is not the same for all the HPV types. As you can see, HPV 16 is detected by more than 90% of all the assays, whereas, for instance, HPV 31 and 39 or 68 has a very low sensitivity by some assays. And you can also see that in 2010, the green bars are for 2010 and the purple bars are from 2014. And the sensitivity reported by the laboratories has increased for almost all HPV types. So by 2014, most of the results reported close to 90% sensitivity by HPV type. Like I said, we have been issuing these proficiency panels during 6 years. We are also made the last issue in 2017. We don't have the data from that yet, but we can already see is that the laboratories that has been participating more than 1 year are improving. You can see here that in 2008 it was 30% only that were 100% proficient, whereas now in 2014 it was 54% that were 100% proficient. And the biggest change here is that in 2008 it was almost half, 44%, that was not proficient, meaning that they had more than one false positive result, compared to that 2014 when it was only 20%. And like I said, the important thing that has improved is the number of false positive tests reported. And it has increased from in 2008, it was 41% of the laboratories that had no false positive result. And in 2014, it was 72. This is still rather low. It should be, I think, close to 100 that has not false positive results. And it seems to be that a lot of people still have actually more than 3 positive results in each sample. And these are laboratories that has performed HPV testing for more than 6 years. So as a conclusion, in proficiency panel testing, the internationally standardized evaluation of performance using both a method and laboratory, using blinded samples traceable to international standards, has successfully been issued regularly. A widely used method with good performance is linear array. There is also a good alternative that is an in-house method that we call Pygmy Tube that was established by the WHO HPV LabNet. This is an acceptable non-commercial alternative. There are several methods with proficient result that is used by one or a few laboratories, which makes it difficult to make a conclusion of the performance of the test. Adequate performance in HPV detection and typing evaluated in international standard proficiency testing will enable meaningful compilation of HPV data from different laboratories. Thank you for your attention.